In this video, we will go over how to freeze cultured suspension or non-adherent cells. Remove cultured suspension cells from the 37-degree incubator. Using a pipette, resuspend the cell suspension to prepare cells for counting. Count cells. Prepare cryogenic vials that will be used for freezing of the cells and conical tubes in which the cell suspension will be centrifuged. In our instance, we are using 4 conical tubes. Spin the cells at 300 G for 5 minutes. After spinning, carefully aspirate the culture medium, being careful not to disturb the cell pellet. Resuspend the cell pellet in 4 degrees Celsius cell freezing medium at a density of approximately 2 million cells per milliliter. Dispense 1 milliliter cell aliquots into cryogenic vials. Place vials in an insulated container for slow cooling and store at -80 degrees Celsius overnight. Transfer vials to liquid nitrogen the next day for storage.
For more products and services to support your drug research and discovery efforts, visit bpsbioscience.com.